Hi everybody. In this video, we're going to talk about the structure of DNA. And to be precise, we're going to talk about how to make the structure of DNA. But before I'll start off with this topic, I want to give you a little introduction. I'm from BBLC, that is Brain Based Learning Center. And on this platform, we are putting videos regarding the syllabus of biology of +1 and +2. So if you are about to take your board exams or your entrance exam, these videos are very beneficial for you. So please do take the benefit of these videos, and we're going to cover all the syllabus on these videos. And please do recommend these videos to people who need this, right? And if you want to go for the full course, you can go to our social media platform and the link is available over there, and you can click onto the link and you can enroll to the full course. So this is about BBLC. Now let's move on to the topic of how to make a structure of DNA. So let's begin with the formation of DNA structure, right? So let me write it. Structure of DNA. We are going to talk about the structure of DNA. So, our human body is made up of cells. We all know this, right? For example, when we have to make a house, we need bricks, right? And with so many bricks, we are making a house. Similarly, our body is made up of cells. Millions and millions of cells are required to make our whole human body. And the cells in our human body is eukaryotic cell. And if we are saying eukaryotic cell, that means we have cells in which nucleus is present, right? So in nucleus, what we have a genetic material. So DNA is basically what? It is a genetic material, right? So genetic material. So what we have to talk today is the genetic material which is present in the cell, in nucleus. It is present, that is genetic material. So let's make the diagram of eukaryotic cell, in which today I'm just gonna make the nucleus, right? There are other things also present in this, but I'm gonna just draw the nucleus. Okay, so let me draw the structure of a cell. So this is an animal cell. All right. So now in this we have nucleus. So this is a nucleus. And in nucleus what we have? We have genetic material, that is DNA. So let's say this is a genetic material, that is DNA. So this is a cell. I'm not drawing cell wall, right? Because this is a eukaryotic cell. So this is a structure of a cell, and this is nucleus in this, and this in the nucleus we have a DNA, and today we're going to talk about this DNA, that is a genetic material. So what is the full form of DNA? That is deoxyribonucleic acid, right? So what is basically DNA? DNA, in DNA we have all the information. In other words, we can say DNA is the brain of a cell, right? So in DNA we have all the information, right? So for example, whatever DNA is present in a cell, it has all the information. But what happened? The DNA which is present, from DNA, what is made? That is RNA, and from RNA what is made? That is protein. So whatever DNA is active in any particular cell, that protein it will make, right? So what I'm basically saying, from DNA RNA is created, from RNA proteins are created. So as I'm saying that DNA is a brain of a cell, right? So in every cell, every information is present, but only that information will be acted upon which particular information is activated. For example, what I'm saying, in pancreas, that DNA is activated, which will say that insulin need to be secreted from that particular cells. But that same DNA is also present in some other organ. Let's say it is present in stomach also, right? In stomach that gene is also present, but that gene is not activated in the area of stomach. So in stomach insulin is not produced, right? But in the pancreas, this insulin gene is activated, so it is secreted in pancreas only. So what I'm trying to say, the whole information is present in the genetic material, but only those information is released in the cell which is activated at that point. So basically I'm saying that whatever DNA will say, that protein will be produced, and that will be activated in that particular cell. So in short, we can say DNA is a brain of a cell, right? Brain of a cell, and all the information is present in the DNA. All right. So we call it a genetic material. All right. So this is just an overview of DNA, that is a genetic material. So what is genetic material? The full form of genetic material DNA is deoxyribonucleic acid, right? And from DNA, RNA is produced. RNA is ribonucleic acid, and from RNA, proteins are produced. So in near chapters, we're going to talk about how DNA will replicate, and how from DNA, RNA is prepared, and from RNA how proteins are prepared. We're going to talk about. For example, from every cell there is a replication of a further cell. For example, from this particular cell, if replication will occur, so when replication will occur, two cells will be produced. So when two cells will be produced in nucleus, the RNA will further produce, right? DNA will further produce. So this we will talk about. So we will talk about all these things, right? But in this video, what we're going to talk about, the structure of DNA, that is a genetic material. So this is the very basic of DNA, that what it is, what is the information DNA has. So all this. Now let's talk about the structure of DNA in precise, right? All right. So now we're going to talk about the structure of DNA. So first thing, we need to understand that DNA is a polymer, right? DNA is a polymer. Now what is the meaning of polymer? Polymer is something which is made up of small unit, or you can say chain which is made up of small units is known as polymer, right? And that small unit is known as monomer, right? So if we are saying DNA is a polymer, that means we are saying that DNA is made up of a small unit. DNA is a chain of some small units like this, right? And this small unit, one unit of this is known as monomer, right? So there are other things are also present in our human body, which is a polymer. For example, protein is a polymer, right? And what is the monomer of protein? Amino acid. So one protein is made up of so many amino acids. The chain of, in other words, we can say the chain of amino acid is known as protein, right? And other things are also there. For example, we can say carbohydrates, right? So that is also a polymer, right? So there are so many things in a human body which is a polymer, right? So similarly, we are saying that DNA is a polymer, and that means DNA has small unit monomers, and joining these monomer will become a polymer of DNA. Now we need to understand what is the monomer of DNA. And what is a monomer of DNA? That is nucleotide. Many nucleotides combine making a chain makes a polymer of a DNA, right? So what we are saying, nucleotide will join together and form a polymer of DNA. So in other words, we can say it is DNA is a polynucleotide. Can we say that? DNA is a polynucleotide. Okay, so now to understand the structure of DNA, what we need to understand? We need to understand the structure of monomer, right? So we need to understand the structure of monomer, that is nucleotide. So let's begin and talk about the structure of monomer, that is nucleotide. So now we're going to talk about the first we need to understand the structure of nucleotide. So let's talk about the monomer nucleotide. So now we need to understand nucleotide, which is a monomer of DNA. Let me write this also. This is a monomer of DNA. It is made up of three things, right? Nucleotide is basically made up of three things. So what are those things? Number one is nitrogenous base, right? Number one is a nitrogenous base. If we need to make a nucleotide, first thing what we need is a nitrogenous base. Now what is a nitrogenous base? Nitrogenous base is an organic compound. These are organic compound which is made up of carbon chains. Definitely you know this. If it is an organic compound, it is made up of a carbon chain. So further, nitrogenous base is of two types, right? So if it is a carbon nitrogen one ring, it is known as pyrimidine. If it is made up of carbon nitrogen two rings, then it is known as purines, right? So I have made two rings, so it is known as purines. And if it is made up of one ring, it is known as pyrimidine, right? Again, what we are talking about? We are talking about one monomer of DNA, right? That is a nucleotide, right? So nucleotide is further made up of three things, right? One nucleotide need three things so that we can make a structure of a nucleotide. What is those things? Number one is a nitrogenous base, right? And nitrogenous base is what? It is an organic compound, and organic compounds are made up of carbon. So further, nitrogenous bases of two types, right? If it is a carbon nitrogen two rings, then it is known as purines, right? And if it is made up of one ring, it is known as pyrimidine, right? So this is a pyrimidine. So further, purines are of two types, right? That is adenine, and second one is guanine, right? And further, pyrimidine is of two types. First is thymine and second is cytosine, right? So this is the first thing that is required to make a nucleotide, that is nitrogenous base, right? So this is done. Now let's talk about the second one. So second thing we need to make to form a nucleotide is pentose sugar, right? Second thing to make a nucleotide is a pentose sugar. So we know this, all the sugars are what? These are carbohydrates. All kind of sugars are carbohydrates, right? So let me draw a structure of a pentose sugar here. So this is oxygen, and this is, let me draw it again. All right. So I'm making a structure of pentose sugar, right? This is a ring structure of a pentose sugar. Pentose sugar looks like this. There's no smiley in this. I just make it. So this is a oxygen, and this ring type structure is of pentose sugar. So this is just a simple structure of a pentose sugar. Now come back what we are talking about. So we are talking that second thing required to make a nucleotide monomer of DNA is pentose sugar. And all the sugars are carbohydrates. We all know this, right? So carbohydrates are further classified into three things, right? Three. And what is the classification? This classification is based on the number of carbon chain, right? So first is a monosaccharide. Second is a oligosaccharide. And the third one is a polysaccharide. Third is a polysaccharide. So I have already told you that this classification is based upon the number of carbon chains, right? So in case of the first one, if we are talking about, in this we have C3 to C7, right? So number of carbons which are present in monosaccharide will be C3 chain and till the C7 chain, right? In monosaccharide there will be nothing like C2, right? You need to remember this, and it will not go beyond C7. So C3, from C3 to C7 is a monosaccharide. And in case of oligosaccharide, so from 2 to 9 it is oligosaccharide, and if it is more than 10, right, more than 10 it is polysaccharide. So what I'm saying basically, if it is a chain of more than 10 like this, so if it is a chain like this, it is known as polysaccharide, and between 2 to 9 it is oligosaccharide, and if it is and carbon from C3 to C7 it is monosaccharide. Now if it is C3, it will be known as triose sugar, right? If it is C4, it will be known as tetrose. If it is 5, it is known as pentose sugar. If it is C6, it is known as hexose sugar. If it is C7, it is known as heptose sugar. Now, what we are talking about, what we are trying to find out, it is pentose sugar. So now we can see it is a pentose sugar. Why I have told you regarding all these sugars? Because we need to understand that from where this pentose sugar is coming, right? We need to understand this. So now you have a little clarity regarding the second thing of this monomer that we need to make a nucleotide, that is pentose sugar, that is a chain of 5 carbon. Is it clear to you all? So second thing is a pentose sugar we need to make a nucleotide. Now we need to understand little bit more about the structure of pentose sugar. So let's first try to understand that. All right. Now what we said that now what we are doing, we are trying to understand the structure of pentose sugar, right? So we just said that pentose sugar is made up of 5 carbon, right? So this 5 carbon we make. So on carbon number 1, aldehyde group is there, right? So here we have aldehyde group. And then we on the second carbon we have OH group. Third carbon also we have OH group. Fourth carbon also we have OH group. And the last one also we have OH group, right? And now, so this is a chain structure of pentose sugar, right? This is a chain structure. But we don't need a chain structure. We need a ring structure. So we need to convert this chain structure into a ring structure. So what will happen? So this is a C1, then we have a C2, then we have a C3, C4 and C5. Now something will happen between C1 and C4. What will happen? C4 will take this OH and will go to this place, right? And from that, this H will make a bond with this O and this O will make a bond with this C. So this will be a bond, right? So what I'm actually saying right now, so this OH will be removed from here and it will go here, right? This this OH, from OH, H will go here and one O is left, right? So let me make it again. So what I'm saying basically, here was OH and OH will go here, right? And this O will make a bond with this. I guess it is removed. So let me draw it again, the structure of pentose sugar. So what we said, there are total 5 carbons, right? And here we have a aldehyde group, and then we have alcohol here also, right? Okay. So this is a chain structure, right? So we don't need chain structure, we need a ring structure. So I said something will happen in first carbon and the fourth carbon. Now what will happen? This OH will be removed from here and it will make a bond with the C1, right? So it will be OH. Now it will make a bond with this H, and this bond will be removed from here because this C will make a 4 bonds, right? 1, 2, 3 and 4. Now 4 bonds of this C is complete, as it is making this H is making a bond with this O, right? Now this O will make a bond with this, right? So what will happen? This O will make a bond with this. Now it will be like, so the whole story will be like this. So now this whole will make a ring structure, right? This whole will make a ring structure, and C5 will be standing outside of this. So now let me draw it to make you understand how it will become a ring structure, right? So how it will be a ring structure? Now this is the oxygen. Now this is C1, C2, C3 and C4. Now the C5 will be here. You can see here that C5 is standing, is like a hanging outside, right? So on this, now in this area you can see, in this area this is a C1 carbon and here it is C1 carbon. Now the C made a bond with this and there is a OH group, right? So here we can put a OH group, right? OH, right? And one H is there, and here also we have OH group, right? And H is also there. Here also we have OH group and H is there. Here also we have H. And on C5 we have CH2OH, right? So this is C1, this is C2, this is C3, this is C4 and this is C5. So this is the ring structure of a pentose sugar. So we need this to make a nucleotide. This is a pentose sugar, right? I hope this ring structure is clear to everybody. So we need this ring structure to make a one monomer of nucleotide. So now we are done with the ring structure of a pentose sugar. Now what we have to do, we first talk about the nitrogenous base, right? Now what we need to do, we need to attach nitrogenous base with this pentose sugar, right? Now one thing in the previous case I need to tell you, I forgot. So we talk about the pentose sugar. We understand that under carbohydrates we have monosaccharides, and under monosaccharide we have a C5, that is a pentose sugar, right? So the example of pentose is, number one is ribose and number two is deoxyribose, right? So to make the diagram, so make the structure of DNA, we need what we need? We need deoxyribose. As the full form of DNA is what? That is deoxyribonucleic acid, right? So nucleic acid. So what we need? We need the second one, that is deoxyribose. So deoxy means we need to remove the oxygen, right? So if we'll remove the oxygen from this ribose, this the structure what we made. So if we'll remove oxygen from this, it will be deoxyribose, right? So right now this is a structure of a ribose, right? This is a structure of a ribose sugar. But we need deoxyribose to make the structure of nucleotide. So we need to remove one oxygen from this ribose sugar. So what we will do, right? So we will remove the oxygen from this C2 carbon, right? we're going to remove one oxygen from this C2 carbon, and it will become what? It will become deoxyribose. So let's remove the oxygen. We are removing oxygen from carbon number 2. Only H will remain here. Now this structure is known as a deoxyribose sugar, right? Now the next thing we were talking about, now to make the real structure of nucleotide, what we need to join nitrogenous base, the first thing we talk about, and the second is the pentose sugar, that is deoxyribose sugar. We need to combine these two, right? And then we need to join the last thing, that is a uh, the last structure which we didn't talk till now. So let's first join these two, that is first one, that is nitrogenous base, and the second is the pentose sugar, that is uh, deoxyribose sugar. So let's join these two. All right, now what we are doing, let's make the structure of pentose sugar first. And so this is the structure of pentose sugar. And this is the last one, CH2OH, right? So here we have OH group, and here also we have OH group. Oh, sorry, we have H only, right? Here we have H, and here we have OH, and this is CH2OH, right? Okay. Now this is a deoxyribose sugar. Now first thing which we need to join it with, that is nitrogenous base, right? So we already know nitrogenous base. Regarding nitrogenous base we talk about. So nitrogenous base we have two, that is first thing we talk about is the purine, and second we talk about is the pyrimidine, right? So under first purine we talk about adenine and guanine, and second in pyrimidine we talk about what? That is thymine and cytosine. So these are the nitrogenous base. So we can attach anything, right? We can attach adenine, guanine, thymine, cytosine, whatever, to make the nucleotide. So let's join one. So let's talk about the adenine. Let's say we are joining a adenine. So what we have to do, we have to remove this OH from here, and we need to attach adenine at this place. So at C1, we're gonna remove OH, and here we're gonna add a nitrogenous base, that is adenine. So here we are going to attach adenine here, right? And the bond which sugar, which carbohydrate will make with the adenine, with the nitrogenous base, is what is known as a glycosidic bond, right? So this bond is known as a glycosidic bond. You need to remember this. The carbohydrate, the characteristic bond of carbohydrates is known as glycosidic bond, right? Is it clear to you? So this is known as a glycosidic bond. So on C1, we put a nitrogenous base, that is of adenine, right? Okay. So now this structure, when we add nitrogenous base with the sugar, with the pentose sugar, this structure is known as what? This is till now, because the third thing we didn't attach till now, we didn't add in this structure. So till now we are not reached to make a full monomer of nucleotide. So we are just with the two things, right? And to make the whole complete structure of nucleotide, we need three things. I hope you remember, right? In the previous slide, we talk about that to make the nucleotide, we need three things. But till now, we just talk about two things. One is the nitrogenous base and second is the pentose sugar, right? So when we are joining nitrogenous base and a pentose sugar, the structure comes up is known as nucleoside, right? So what is the name of the structure? Till now what we made, that is nucleoside. But what we need to make? We need to make a nucleotide, right? When we'll make a nucleotide, then the one monomer of DNA will be completed. Now let's talk about the third thing. The third thing, we need to complete the structure of a DNA monomer, that is nucleotide, is a phosphate, right? So we need phosphate. So first phosphate is what? That is a phosphoric acid. Okay, so basically it is phosphoric acid. What is phosphoric acid? Phosphoric acid is H3PO4, right? So now this is the third thing which we need to add in to complete our structure of a nucleotide, right? So now let's add the third thing, that is phosphoric acid, that is H3PO4. Now we need to add. Now where we need to add this, that I'll tell you. But before, let's talk about the structure of H3PO4. So let's open this structure. I'm doing it here. So the open structure of, let's do it on the next slide. Okay. So we are talking about the phosphoric acid, right? And what is phosphoric acid? That is H3PO4. Now open this structure. P is 1 and we have 3H, right? And we have 4O. We have 4O, OH, OH, OH and 1 O. So O need to complete the bond and O make two bonds, right? So now our structure is complete of H3PO4. Now we need to add this structure, this phosphate group to our nucleoside, and then our whole nucleoside will complete and form a nucleotide. So now let's add that. So I'm making a structure again here. Now this is the C5, right? So we have attached adenine here, and here we have only H, and here we have OH, right? So here it is CH2OH, right? Now this phosphate group we gonna add at C5, right? So we need to add this whole group on C5. Let's open C5 also. So I'm opening here. So what we are adding here, that is CH2OH, right? So this is a fifth carbon on and on fifth carbon this phosphate group will be we are joining this phosphoric group, right? So this is C5. So we said it is CH2OH, right? This is OH. Now here we have to add this phosphate group. So this is All right. So what we need to do now, when there is a when this is a C5, right, and we have a uh, phosphoric acid. So it is phosphoric acid is inorganic compound, right? It is acid, but it is a inorganic compound, right? So the basic chemistry we all know that OH is a alcohol, right? And CHO is a aldehyde, COOH is a carboxylic acid, right? So we all know about this, right? So now what we need to do, we need to attach phosphoric group with this C5, that is a carbon, right? So now what we are doing, when these two will, when this group will attach with this group, there will be a formation of a bond, right? And that bond is known as a ester bond, right? And how, when ester bond is formed, what is released? Water is released. And what is water? That is HOH. So now from this area we need to remove water, we need to remove HOH. So let's remove HOH from this area and form a ester bond. Okay, so now we need to remove HOH. So from here it is removed and only this will remain, right? And this bond is known as what? This bond is known as ester bond. And as it is a formation between a phosphoric group and C5, this bond is known as phosphoester bond, right? So you need to remember this. And what was the bond which is made between a nitrogenous base and the carbon one, C1? What is the name of that bond? We just talked in the previous slide. What was the name of that bond which is formed between C1 and the nitrogen base? What was it? It is known as a glycosidic bond. We need to remember this, right? So glycosidic bond. This is a characteristic bond of a, right? This is a characteristic bond which is made between carbohydrates. So this is a glycosidic bond. Now this whole structure is known as nucleoside. Oh, sorry, nucleotide. Now this is one monomer of DNA, right? This is a monomer of DNA. And this is what we are trying to make, right? So what we are trying to make, we are trying to make the monomer of DNA, which is nucleotide. So this is all about what we are trying to do, right? Now let's talk about recapitulation of this. So what we are trying to do, we start with what? We are trying to make the DNA structure of DNA, right? So what we did, we started from the very beginning that we have a eukaryotic cell, and in every eukaryotic cell what we have a nucleus, right? And in every nucleus what we have a DNA. So we start from there and we need to study the structure of a DNA. And we come to know that DNA is a polymer, right? And what is polymer? If we are making a chain with small unit, and that chain of small unit is known as a polymer, and that one small unit of that chain is known as monomer, right? And what is the monomer of DNA? That is nucleotide, right? So we need to understand the structure of a nucleotide first, so that we can make the chain of a DNA. So in this we talk about the structure of monomer, that is nucleotide, right? And then we come to know that to make the structure of nucleotide, what we need three things, right? And you can say, if you want to make some kind of a recipe, then we need these ingredients, right? So similarly, to make the recipe of nucleotide, we need three ingredients. And what are those ingredients? Number one is nitrogenous base. So we talk about this. Nitrogenous base is what? That is a organic compound, right? It is made up of carbon rings, and further nucleotide nitrogenous base is divided into two, that is purines and pyrimidine. And purines are of further two types, that is adenine and guanine, right? And then pyrimidine is again further two types, that is thymine and cytosine. This is about the nitrogenous base. And second component which we require to make the structure of one monomer nucleotide is pentose sugar. Then we come to know about all the sugars are carbohydrates. Then we talk about the three type classification of carbohydrates, and then we come to understand that pentose sugar comes under monosaccharide, right? Then we talk about the structure of a pentose sugar, and then one example of pentose sugar is a D that is a deoxyribonucleic acid. And deoxyribonucleic acid, we need that in a DNA, right? So now we understand the structure of second thing, that is a deoxyribonucleic acid. Then we join the nitrogenous base with the deoxyribo sugar, and then we made the structure that comes out to be a nucleoside. Now you need to remember, this is a question of a NEET, right? Your entrance exam. They may ask you what is the difference between a nucleotide and a nucleoside. So you need to understand nucleoside is what? When only nitrogenous base and your sugar is attached. That is known as a nucleoside. But if phosphate group will be added in that, then it will become a complete structure of a nucleoside. So this is a one question of a NEET exam, your entrance exam, right? So in the last, we talk about the phosphoric acid, and we attach that phosphoric acid with the nitrogen base and our sugar, right? That is a pentose sugar, and then our whole structure of nucleotide is complete. So this is about one monomer, that is nucleotide. And in the next video, we're going to talk about how to form the chain of a nucleotide and form a full polymer of a DNA. I hope this is clear to you. If you guys have any query regarding the structure of monomer, you can keep the comment. You can drop the comment in the comment box, and you can ask the question if you have any query. So thank you so much for watching this video.